it is known as 30 nanometer fibers. And then we may loop these and we may arrange them around about some protein structures to form something that has been called rosettes, and then we could further coil these things into what we ultimately refer to as a chromatid. Okay, so chromatid is a DNA molecule, and when we have replication of the DNA, we will have another chromatid, and this is then a complete chromosome. Okay, so as I said, not all of these levels are completely understood, so I will share with you some basics of this process, starting with the formation of nucleosomes. So nucleosomes, as I said, are composed of DNA and protein core. Yes, please.
So is that coiling structure proposed, or is it known?
Which one?
The one you just showed. Is that just proposed, if it's not fully understood, or do we know that's the structure, we just don't know how it's formed?
Exactly.
Okay.
Okay. So it's not fully understood how we form these things and what the molecular basis necessarily is in all these cases. Okay. But we know, we pretty much understand these structures. Okay. I'm not quite sure whether, for example, it is understood whether or not there is a certain, and I'm just speculating here, but I'm not quite sure whether it's known, for example, whether or not there is a certain machinery necessary to form certain levels here or not, or whether these are self-condensing or whatever you want to call it. Okay, and I'm not fully aware of that also. But for sure, we don't know all the details of these processes. Okay, so what we know is, for example, that in nucleosomes we have stretches of DNA wrapped about a protein core that is formed by so-called histones, in particular 2 copies of histone 2HA, 2 copies of 2HB, 2 copies of H3, and 2 copies of H4. Okay, so we have a total of 8 proteins, and then we have a stretch of DNA that is 146 base pairs long that wraps about the histone, the protein core about 1.7 times. Okay, I think in the textbook it says 1.65 times or something. Never mind, it's 146 base pairs long. And this condenses DNA to a certain factor. Never mind. Now, by definition, a nucleosome not only contains this 146 base pair long part that is wrapped in a left-handed supercoil, actually, about this protein core, but it also includes the linker, a stretch of DNA that is 54 base pairs long that links this part to the next DNA that is wrapped about a protein core. Okay, so by definition, a nucleosome contains DNA that is 200 base pairs in length, wrapped about our protein core formed by H2A, H2B, H3, and H4. And then we'll see on different representation that there is another type of histone, H1, which actually attaches more to the outside of this nucleosome. Okay. So obviously, if we look at this schematic representation, it's comparable to beads on a string, okay, and this can actually be visualized on experimental conditions if we treat DNA with low ionic strength solutions. And then on the microscope, electron microscope, we can actually see these structures. Now, I want to caution you, we will see in the next couple of hours that we have here together, we'll see a number of pictures that look just like that, okay? But they actually will show different things. Okay, so in one case, for example, we will see lots of ribosomes attached to an mRNA synthesizing protein that looks pretty much just like that. Okay, but it doesn't show in all these cases the same thing. Okay, so here we are talking about nucleosome structure. Okay, for the condensation of DNA. but it looks like beads on a string. Okay, so don't think that I always show the same picture. Like, these different pictures showing different things actually. Okay, so this slide just shows that then we may need topoisomerases to remove conformational strain. Never mind, it's not such great importance. What is of greater importance is to take a look at this table to understand some structural features of the proteins that actually participate in this condensation process here, namely histones. Okay, so in particular, histones H2A, H2B, H3, H4 are highly conserved proteins. That means that they haven't really mutated a lot or changed a lot if we compare the histone sequences and histone structures of organisms that from a evolutionary point of view are quite distant. Okay, and I think your textbook gives an example comparing certain histones in a cow with histones in a pea or something like this, and textbook says, okay, there are only 2 amino acid residues mutated. Now, I certainly don't want you to remember a cow or pea or whatever, but I want you to understand the importance of the fact that these are so highly conserved, these proteins, namely that it shows that these proteins are well tuned for what they are supposed to be doing, and if we have a high degree of conservation of biomacromolecules, it means usually also that if you mess with the structure, then it has a detrimental effect on the organism, and then it doesn't survive. Okay, so these structural features of histones that have evolved over a long time are so well tuned that, okay, changing them is probably not a good idea for the survival of the animal or organism in general. Okay. So for H1, it's not quite that extreme. It's not as highly conserved because it actually attaches to the outside, so the role is probably not as important as the other one. It certainly is very important, but it allows more variation, obviously. If we look at the molecular weight of these types of histones, overall they're relatively small. Okay, H1 seems to be a little larger. But what I want to point out is that these histones all contain quite a bit of positively charged, meaning basic amino acid residues. Why would that make sense? Because these proteins interact with DNA, which is negatively charged in the sugar phosphate backbone, and we have interactions a lot via charge-charge interactions. Okay. So it is known, and there's a section in your textbook on that is quite extensive. It is known that the interaction between histones and DNA doesn't appear to be random, okay, but occurs in certain sequences, for example, that are rich in A and T, okay, and your textbook can give you more information there. What I would like to say, and this is a very oversimplified statement that I make here, is that the interaction between histones and DNA can be modified by modifying amino acid residues in the histones. Okay, and there's a great variety of different possibilities how these amino acid residues can be modified, and they just listed a couple here, namely what is quite common is acetylation or methylation, but also phosphorylation, which then affect the interaction of these proteins with DNA, okay, and may affect how tightly condensed DNA is, and this affects then also processes like transcription. Okay, so certain modifications, certain organisms may actually facilitate transcription or make it more difficult for transcription. But this is a quite complex topic. I'm certainly not an expert there. It's also, I think, not particularly well understood in some cases. But if you hear about methylation of histones and things like that. So let's come to the next level. So I told you that the nucleosomes can then be wound in a certain way to assume these 30 nanometer structures which are called 30 nanometer fibers because they're 30 nanometers wide cross section, and they contain approximately 6 nucleosomes. And this confers another level, another extent of condensation. Here just added some numbers that I found somewhere, a factor of about 100 or so. And ultimately, of course, you have to have a condensation factor that brings your length, let's say that is in the centimeter range, down to microns or something like this. Okay? So you can calculate yourself what it is from centimeter to microns. Okay, so then you find that in textbooks also that these fibers, these 30 nanometer fibers, can be looped, and then these loop structures may be arranged about scaffolds that are formed by proteins. There are a number of proteins that have been identified as participating in this kind of arrangement, condensins, never mind. What you also see here is that certain genes that usually need to be expressed at the same time, like histone genes. Okay, if we need to express histones, then we not only need H1, but we also need H2A, H2B, H3, and H4. So it would make sense to actually express these proteins at about the same time. And it has been found that often enough appears that genes that need to be expressed at around the same time are found in the same areas of such loops. This is another picture that I found somewhere which shows some of the levels that we discussed here. We also see a new term here, namely the nucleosome to which the H1 histone has attached. They call it chromatosome. Okay, never mind. And then they don't really point out this nuclear scaffolding and these rosettes that we saw previously, but they just say, okay, we form then loops that are 300 nanometers in length, and then we form certain fibers with this, and so on, up to the level of chromatids and making chromosomes which are composed of 2 chromosomes. Okay, so now that we have some understanding of DNA, we can talk about processes that involve DNA, like transcription, translation, potentially replication. So this slide here shows something that has been known and is known as the central dogma of molecular biology, which was first formulated by Francis Crick, one of the discoverers of the Watson-Crick double helical structure of DNA in the 1950s. Okay, and what Francis Crick actually pretty much put in words was that DNA, okay, he was the discoverer of the double helix, okay, DNA pretty much contains the information not only for its own replication, okay, and we talked about this just very briefly. If we have complementary strands, we can take one as a template for the production of a copy of the complementary strand and vice versa. Okay, so now that we know the structure, that's a no-brainer, right? But then he also realized, okay, DNA also serves as a information storage molecule to store the information for other biological products, like RNA, okay, and also protein. So DNA, if we go all the way to a protein here, DNA has to be first transcribed into a certain form of RNA known as messenger RNA. So a gene, and we saw this in the schematic representation last time, in the process called transcription, an RNA copy of a gene is made, okay, by RNA polymerase, and then this information is utilized by ribosomes to translate it into a corresponding amino acid sequence. And the amino acid sequence, the peptide, will then fold in a particular way, assume a certain three-dimensional structure, and we may hopefully get a functional, biologically active protein. Okay, so these two steps that we have to take in order to come from the DNA level to the protein level have been called transcription, because first we transcribe the information from the DNA to a corresponding RNA sequence, and then we translate this information from a nucleic acid, namely a messenger RNA, into a protein. Okay, so nowadays we also know that we can also go from RNA back to DNA with reverse transcriptases. So if you have retroviruses which store their genetic information in the form of RNA, reverse transcriptases would be utilized in a host organism then to actually convert the RNA information to DNA, and then we would express proteins, virus capsid proteins, for example. Okay. So, due to the, don't even remember how many, but I think 2, possibly 3 snow days that we had, I'm not quite sure whether or not we will be able to actually talk about DNA replication. I'm relatively hopeful, but if not, then not. There are years where I have time to talk about DNA replication. There are years where I don't have time, so it's not a big loss. Okay, it's relatively straightforward anyway. Most of you have heard about this anyway. Also, I doubt that we will have time to talk in more detail about interaction of proteins with nucleic acids. I pointed this out last time, which you find in your syllabus as the last item to be discussed. I not usually have time. We'll talk about transcription regulation, the absolute fundamentals, starting today or Tuesday. But what we are going to discuss is transcription and translation. So, and for this, we have to revisit pretty much the definition of a gene. So, okay, I mentioned that genes encode the information for functional biological products, and I said for simplicity we'll just assume that this usually is a protein, but also RNAs. The information for RNAs is encoded in DNA, okay, in genes. So the definition of a gene has actually changed quite a bit over the years. Okay, and you may think, oh, I know what a gene is. No, you don't. Okay, because it's a matter of definition, how you define a gene. Okay, and still the scientific community has problems really agreeing on one definition for a gene. Okay, so originally genes were defined as being responsible for causing a certain phenotype, visible property. And this is based on Mendel's experiments with peas pretty much in the 1850s and early 1860s, which were forgotten for quite some time, but then were rediscovered in the early 1900s. And around that time, a Danish scientist, actually, botanist if I'm not mistaken, Wilhelm Johansen, actually defined pretty much genes as something that causes a certain phenotype. Okay? And what you have to understand is that at that time, no one knew that hereditary information is actually stored in nucleic acids. Okay, so again, you have to put yourself into the shoes of people living at the time, and we just take things that have been discovered before us actually as granted. Okay, but when Mendel did his experiments crossing certain peas and counting peas, and I just saw yesterday, I looked this up again, tens of thousands of experiments that he performed in a relatively short period of time. He had no clue what actually caused a pea shape to be round or not round, or have flowers having a certain color or different color. He didn't know that. Okay? For the longest time, and again, just think about it. For the longest time, scientists thought that this type of information, hereditary information, is actually stored in proteins, not nucleic acids. Okay. Only in the 1940s, Oswald Avery actually performed experiments that showed that genetic information is actually stored in nucleic acids, and this was confirmed in the 1950s. Okay. But up to that point, we're talking around this time where the double helical structure of DNA was discovered. Remember, this was in 1953. People didn't know that. Okay. So, strictly speaking, when Watson and Crick actually tried to elucidate the structure, the double-stranded structure of DNA, or the structure of DNA, they didn't even know for sure that it would actually carry genetic information. Okay, so that's something you may want to keep in mind. In the 1940s, however, then a lot of experiments were performed, and